Today we're talking about Brucella, one of the most important zoonotic bacterial genera in veterinary medicine. These organisms are stealthy, persistent, and incredibly well adapted to their hosts. Brucella species cause reproductive losses in livestock, chronic infections in wildlife, and serious disease in people. They're also a major One Health priority because of their economic impact and zoonotic potential. By the end of this session, you'll have a clear understanding of the major Brucella species, how they cause disease, how we diagnose them, and how we prevent transmission across species. Let's get to it. Are small gram-negative Coccobacilli. They're slow-growing and facultative intracellular pathogens, meaning they survive and replicate inside macrophages. That intracellular lifestyle is a big part of why they're so persistent and so difficult to eliminate. Each Brucella species is strongly host-adapted, which helps explain the very specific disease patterns we see in different animals. The major Brucella species you'll encounter. B. abortus is a classic cause of bovine brucellosis. B. melitensis is the most pathogenic for humans and is associated with sheep and goats. B. suis affects pigs and feral swine and is a growing concern in the U.S. B. canis causes reproductive disease in dogs and is zoonotic. B. ovis affects rams and causes epididymitis. Marine mammals, Brucella species B. setae and B. pinnipedalius, are increasingly recognized in seals, sea lions, and cetaceans. Brucella is transmitted primarily through reproductive tissues, placenta, fetal fluids, vaginal discharge, and through ingestion of contaminated materials. Milk is a major route of transmission for B. melitensis and B. abortus. Some species, like B. canis, can also be shed in urine. Because the bacteria survive well in moist environments, contaminated pastures, barns, and birthing areas are high-risk zones. Entering the body, Brucella travel to regional lymph nodes and then spread through the bloodstream. Their ability to survive inside macrophages allows them to evade the immune system and establish chronic infection. They have a strong tropism for reproductive tissues, especially the placenta, uterus, epididymis, and mammary gland. This is why abortion, infertility, and orchitis are hallmark signs. B. abortus causes late-term abortions, typically in the last trimester. Cows may retain the placenta, and milk production often drops. Bulls can develop orchitis or epididymitis. Calves born alive may be weak or infected at birth. This disease has major economic impacts, which is why eradication programs have been so important in North America. Melitensis is the most pathogenic Brucella species for humans and a major cause of abortion storms in sheep and goats. It spreads rapidly through flocks, especially during lambing and kidding seasons. Mastitis is common, and milk becomes a major source of transmission. This species is a major public health concern in many parts of the world. In pigs, B. suis causes infertility, abortion, and orchitis. Feral swine are a major reservoir in the United States, especially in the southeast. They can transmit infection to domestic pigs, dogs, and even hunters who handle carcasses. This species is also zoonotic and can cause severe disease in people. B. canis causes reproductive disease in dogs, infertility, abortion, epididymitis, and prostatitis. It spreads through breeding, whelping fluids, and sometimes urine. Kennels and breeding facilities are high-risk environments. It's zoonotic, though human cases are less common than with B. melitensis or B. suis. B. ovis is unique because it primarily affects rams, causing epididymitis and infertility. It is not considered zoonotic, which distinguishes it from the other Brucella species. Species, the clinical picture is remarkably consistent. Reproductive losses, infertility, weak neonates, and chronic infection. Because Brucella can persist for months to years, infected animals often become long-term reservoirs. Serology is the main diagnostic tool. ELISA and agglutination tests are widely used. PCR is increasingly common and can detect Brucella DNA in tissues or fluids. Culture is definitive but slow and requires biosafety precautions because Brucella is a significant laboratory hazard. In cattle, the milk ring test is a classic screening tool. Treatment is generally not recommended in livestock because Brucella is difficult to eliminate and animals remain chronic carriers. In dogs, long-term combination antibiotic therapy may reduce clinical signs but often does not fully clear infection. Management focuses on preventing transmission rather than curing infection. Prevention is the cornerstone of Brucella control. Vaccination with RB51 is used in cattle in some regions. Test and cull programs have been highly successful in reducing B. abortus in North America. Biosecurity, especially controlling contact with wildlife reservoirs, is essential. Pasteurization of milk is one of the most effective public health measures ever implemented. Brucellosis in humans causes undulating fever, joint pain, fatigue, and chronic infection. People become infected through contact with reproductive fluids, aborted fetuses, or unpasteurized dairy products. Veterinarians, farmers, abattoir workers, and hunters are at higher risk. This is a classic One Health disease. Controlling it in animals protects human health. Brucella species are host-adapted intracellular pathogens that cause reproductive disease in livestock, wildlife, and companion animals, and serious illness in people. Because treatment is difficult and chronic infection is common, prevention and biosecurity are the keys to control. Understanding the ecology, transmission routes, and species-specific disease patterns gives you a strong foundation for managing this important zoonotic pathogen. If you found this video helpful, please like and subscribe. Thanks for joining, and I'll see you next time.